A single vial can protect millions of people, yet it starts as nothing more than genetic code on a computer screen. An mRNA vaccine teaches your own cells to become the factory, but the instructions dissolve within days of doing their job. A flu shot is grown, one dose at a time, inside chicken eggs, and somehow still reaches 1 billion people every single year. A protein vaccine is built the same way your body already knows how to read it, but engineering that shape takes months of trial and error. 3 vaccines, 3 completely different starting materials, 3 timelines that range from weeks to months, and yet every single one ends the same way. A vial, a needle, and an immune system that now knows how to fight back before it's ever been infected. That level of precision shouldn't be possible at this speed or at this scale. 3 completely different starting points, all ending at the same tiny vial. So how do scientists in factories turn raw genetic code, egg proteins, and lab-grown cells into billions of doses trusted to protect the world every single year? Let's step inside and find out. An mRNA vaccine contains no virus at all, not weakened, not killed, not even a fragment of one. It's a set of instructions, and your own body does the manufacturing. It begins in a lab, not a factory floor. Scientists start with the genetic sequence of a single target protein, usually a small piece on the outside of a virus. That sequence is written and stored as data long before it ever becomes a physical substance. Researchers can pull up that sequence from a database, tweak it on a computer, and send the updated design straight to production without ever touching a physical sample of the virus itself. Using that digital blueprint, machines synthesize a strand of messenger RNA, the same molecule your own cells use every day to build proteins. The mRNA is produced through a process called in vitro transcription, where a DNA template guides an enzyme to assemble RNA building blocks in a temperature-controlled reactor. Within hours, a single reaction can generate trillions of RNA copies. But raw mRNA is fragile. It breaks down almost instantly outside a controlled environment. So the strands are immediately mixed with fatty molecules called lipids inside a device that pulses the 2 liquids together at high pressure. This forms lipid nanoparticles, microscopic bubbles roughly 1,000 times smaller than a human hair, each one wrapping and protecting a single strand of mRNA. Engineers control the exact ratio of the 4 lipid types used, because even a small shift in that ratio changes how efficiently the particle is absorbed by cells once injected. The mixture is then filtered, concentrated, and tested for particle size, using lasers to confirm that every nanoparticle falls within a precise, narrow range. Only then is it filled into vials on high-speed lines that can seal thousands of doses per hour, each one automatically inspected for cracks or contamination. The mRNA itself typically lasts only a few days inside your cells, long enough to trigger an immune response, never long enough to alter your DNA. The molecule is designed to disappear once its job is done. Before any of it ships, samples from every batch are tested for sterility, potency, and the exact ratio of RNA to lipid, and a single failed test can mean discarding the entire lot. Cold chain logistics matter just as much as the chemistry. Some formulations need to stay frozen at extremely low temperatures all the way from the factory to the clinic, which is why specialized freezer trucks and packaging have become as important to this platform as the reactors that make the mRNA itself. From a strand of digital code to a sealed vial in a refrigerated case, the entire production cycle can take as little as 6 weeks, a fraction of the time older vaccine methods require, and a large part of why this platform can be redesigned for a new target almost as quickly as updating a piece of software. Chicken eggs, on the other hand, still make up one of the oldest vaccine factories on Earth. Every year, hundreds of millions of flu vaccine doses begin their life inside a fertilized chicken egg, a method that hasn't fundamentally changed in over 70 years. Specialized flocks of hens, raised in controlled pathogen-free facilities, lay eggs used exclusively for vaccine production. These birds live under conditions closer to a hospital than a farm, screened regularly to make sure no outside pathogens contaminate the eggs before they're ever used. The eggs are incubated for about 11 days until they're developed enough to support viral growth, then moved on to the production line. Each egg is candled under bright light to confirm it's viable. Then a small hole is punched through the shell. A tiny amount of the target flu strain is injected directly into the fluid surrounding the growing embryo. The eggs are resealed and incubated again, this time for several days, while the virus replicates by the millions inside that fluid. Once replication peaks, the eggs move to a harvesting line where machines crack them open and extract the virus-laden fluid. That fluid is purified through multiple filtration and centrifugation steps to strip away egg proteins and debris, leaving concentrated virus behind. Some manufacturers spin the fluid through several rounds of high-speed centrifuges, each pass removing a little more of the material the immune system doesn't need to see. The virus is then inactivated using a chemical agent that destroys its ability to cause infection while preserving the surface structures the immune system needs to recognize. Finally, the inactivated virus is standardized to a precise concentration and filled into vials or preloaded syringes. A single egg can produce enough virus for roughly 1 to 2 vaccine doses, which means a season's supply can require well over 100 million eggs, all timed to hatch, inject, and harvest on a rolling weekly schedule. Entire flocks are scheduled months in advance because a shortfall in eggs at the wrong moment can throttle an entire country's supply just as flu season approaches. Some manufacturers keep backup breeding flocks on standby for exactly this reason, ready to scale production up if a strain change or supply disruption threatens the timeline. From a fertilized egg to a sealed syringe, the process takes several months, timed each year to a moving target, predicting which flu strains will circulate before the season even begins, using surveillance data gathered from labs on every continent. Protein-based vaccines skip the egg entirely and instead borrow a trick from brewing. Some of the most widely used vaccines in the world for hepatitis B, HPV, and whooping cough don't use a virus at all. They use bioengineered yeast or insect cells as microscopic assembly lines. Scientists insert a small piece of viral genetic code into the DNA of yeast or insect cells. These modified cells become living factories, each one carrying the instructions to build a single target protein, the same protein that sits on the outside of the actual virus. Once a cell line proves it can reliably produce that protein, it's frozen, banked, and reused as the starting point for every future batch, sometimes for decades at a time. The engineered cells are grown inside massive steel bioreactors, some holding thousands of liters, kept at precise temperature, oxygen, and pH levels for days at a time. As the cells multiply, they continuously produce copies of the target protein, secreting it into the surrounding liquid. Technicians monitor the culture around the clock, adjusting nutrient feed rates so the cells keep dividing without running out of the sugars and amino acids they need to keep producing protein. Once the culture reaches peak protein output, the mixture moves through a purification train. Multiple chromatography columns separate the target protein from cell debris, sugars, and unwanted byproducts, refining the sample step by step until it's nearly pure protein. Technicians then combine the purified protein with an adjuvant, a substance that boosts the immune system's response to it, ensuring the vaccine works even though it contains no living virus. The final formulation is filtered, tested for potency and purity, and filled into vials under sterile cleanroom conditions. A single bioreactor batch, grown from cells too small to see, can eventually be purified and diluted into hundreds of thousands of individual doses, all traced back to one engineered cell line. The same basic method has quietly been running for decades, which is part of why protein subunit vaccines have some of the longest safety track records of any vaccine platform in use today. From a modified strand of DNA inside a yeast cell to a purified adjuvanted dose in a vial, the process can take 4 to 6 months, most of it spent growing and purifying, not manufacturing. Patience, more than speed, is what this method depends on, start to finish. That's how mRNA vaccines, egg-grown flu vaccines, and protein subunit vaccines go from digital code, fertilized eggs, and engineered cells to billions of doses trusted around the world. Precision, biology, and engineering, most of us never think about. 3 completely different starting points. Three completely different timelines, all built around the same goal: teaching the immune system to recognize a threat before it ever causes harm. Until now.